Well, hello everyone. Welcome to Dr. Osman's Edu Trends, swinging to educational trends. Friends, today we will be talking about antibiotics. So, antibiotics are very important class of compounds or organic compounds or medicinal agents. If we try to define antibiotics, we know that antibiotics are chemotherapeutic agents isolated from the microbial metabolites which can kill or abolish the growth of microorganisms. These antibiotics do not cause any harm to the host cell. So, these antibiotics will be killing or abolishing the growth of microorganisms. So, if we try to break the term antibiotic, it is derived from Greek word that is antibiosis. Anti means against and biosis means life. So, it doesn't mean it is against the life of human beings, but it is against the life of the microorganisms which are causing the infection. William was the person who coined antibiotic in the year 1889 while describing the principle of the survival of the fittest. Now, if we move on to the classification of antibiotics, we'll come across different classes. Like the first class will be beta-lactam antibiotics, which includes penicillin, cephalosporins, monobactams and carbapenems. Of course, we will be talking about them in detail when we will go to them in specific. Now, second class will be aminoglycosides, example streptomycin, neomycin, kanamycin, gentamicin. The third class will be of tetracyclines, examples tetracyclines, chlorotetracycline, oxytetracycline, doxycycline, minocycline. We have macrolide antibiotics, erythromycin, clarithromycin, roxithromycin and azithromycin. Then the fifth class will be of lincosamide antibiotics, example lincomycin, clindamycin. And then sixth class will be of polyene antibiotics, example amphotericin B, nystatin, natamycin. The seventh class will be of polypeptide antibiotics, example pesitracin, polymyxin B, colistin, vancomycin, rifampicin. Then we have eighth class, that's the last class, miscellaneous antibiotics, which includes chloramphenicol, novobiocin, mupirocin. Now let us try to decode them in detail. Beta-lactam antibiotics. So in 1929, it was first obtained by fermentation of fungus Penicillium notatum by the scientist, the Scottish biologist and pharmacologist Alexander Fleming. The first naturally occurring penicillin was benzyl penicillin, a benzathine penicillin G. The chemistry of beta-lactam antibiotic is like their structure contains a beta-lactam ring. A lactam is a cyclic amide formerly derived from an amino alkanoic acid. So the Greek prefixes in alphabetical order indicate the ring size. Alpha-lactam antibiotics, which includes 3-atom rings. Beta-lactam includes 4-atom rings. Gamma-lactam includes 5-atom rings. And delta-lactam includes 6-atom rings. So you can see in the picture as well. Now, if we talk about the penicillin. Penicillins has a fused beta-lactam and thiazolidine ring system. You can see in the nucleus, there is a beta-lactam ring and also thiazolidine ring. A free carbonyl acid group, acyl amino side chain, one or more substituted amino side chain. If we talk about the nomenclature, then we'll know that penicillin nomenclature can be done by different systems. The first one is chemical abstract system, that is CAS system. In this system, the numbering will start from sulfur atom and it will be assigned first position, and nitrogen atom is assigned fourth position. And it is called as 6-acyl amino 2,2-dimethyl 3-carboxylic acid. The second system will be United States Pharmacopoeia USP system. In this system, reverse of CA system is adopted, as here the nitrogen atom is given the first position and sulfur atom is assigned the fourth position. And it is called as 4-thia-1-aza bicycloheptane. The third system will be it is considered as a derivative of penam. So unsubstituted bicyclic system with carbonyl group. And the fourth system will be as derivative of penicillinic acid, that is 6-amino penicillinic acid. Penicillins are named as derivatives of penicillinic acid. Ring system with 2,2-dimethyl and carboxyl group as substituent at second and third positions. The fifth system will be trivial system, and carbonyl amino penicillinic acid portion of the molecule is named as penicillin. Trivial system is a simple system serving as a good measure for naming and comparing closely related penicillin structures. Not well suited for compounds having the ring modified derivatives. Now, moving on to the classification of penicillin. Penicillins are classified into the first class, that is natural biosynthetic penicillin, penicillin G, penicillin F, penicillin V and penicillin X. The second class will be semisynthetic penicillins, which includes penicillinase resistance, aryl oxypenicillin, example methicillin, nafcillin. The second subclass is isoxazole penicillin, oxacillin, cloxacillin, dicloxacillin. The second subclass aminopenicillin, ampicillin, amoxicillin. And third subclass is carboxypenicillins, example carboncillin, ticarcillin. The fourth subclass is uridocyclic urease penicillins, example piperacillin, meslocillin, azlocillin. Let us try to decode them in detail. Natural biosynthetic penicillins, example penicillin G, penicillin B, and penicillin X. So you can see on the screen the names of the biosynthetic penicillins and the basic nucleus, the parent nucleus, and substitution is at R position. And you can see when we are replacing that particular R with this all groups, we'll get the different type of penicillins. Along with that, there is an impact name mentioned with a different kind of penicillin, which has penicillin G, penicillin V, penicillin F, 3-pentenyl penicillin, N-heptyl penicillin, which is called as penicillin K, N-pentenyl penicillin, para-hydroxybenzyl penicillin, that is penicillin X. Now moving on to the second subclass, that is semisynthetic penicillin, that is penicillinase resistant penicillins. These penicillins will be overcoming the shortcomings of natural penicillins, where they have poor oral efficiency, sensitivity to penicillinase, narrow spectrum of activity, and hypersensitivity reactions. So we have aryl oxypenicillins, example methicillin and nafcillin. So you can see the structure. Methicillin available with the brand name Sinarcet and IUPAC name 6,2,6-dimethoxybenzamido, 3,3-dimethyl-7-oxo-4-thia, 1-aza-bicycloheptane 2-carboxylic acid. And the second is nafcillin, available with the brand name Nalpen and IUPAC name 6,2-ethoxynaphthalene, 1-aryl-carbonyl amino 3,3-dimethyl-7-oxo-4-thia, 1-aza-bicycloheptane 2-carboxylic acid. Now I would like to say about methicillin is acid sensitivity and has to be injected. Steric shields can be added to penicillins to protect from penicillinase enzyme. Bulky groups on the side chain prevents the penicillinase enzyme to reach the penicillinase active site. Now the second is nafcillin, available with the brand name Nalpen and IUPAC name 6,2-ethoxynaphthalene, 1-aryl-carbonyl amino 3,3-dimethyl-7-oxo-4-thia, 1-aza-bicycloheptane 2-carboxylic acid. Nafcillin sodium is a narrow spectrum beta-lactam antibiotic of the penicillin class. As a beta-lactamase resistant penicillin, it is used to treat infections caused by gram-positive bacteria, in particular species of staphylococci that are resistant to other penicillins. Now moving on to isoxazole penicillins, example oxacillin, cloxacillin, dicloxacillin. Isoxazole penicillins are penicillinase resistant penicillin, and here isoxazole ring act as a steric shield and also electron withdrawing group, giving acid stability to structure. So we have this parent nucleus of isoxazole penicillins and replacement of R with the structure mentioned on the screen, and you will get the different types of isoxazole penicillins. In that we have oxacillin, cloxacillin. Oxacillin available with the brand name Bactrocel, replacement with R and R2 will give the structure, and IUPAC name is mentioned on the screen. Cloxacillin is again available with the brand name Ampiclox, and replacement with R1 is Cl and H is for R2, and IUPAC name is mentioned on the screen. In cloxacillin, the chlorine atom ortho to the position of attachment of the phenyl group or phenyl ring to the isoxazole ring enhances the activity of cloxacillin. Dicloxacillin available with the brand name Diclosil, replacement with 2 chlorine atoms at R1 and R2. The substitution of chlorine atoms on both carbon ortho to position of attachment of phenyl ring to the isoxazole ring is presumed to enhance further the stability of oxacillin, congener dicloxacillin. Now moving on to the second subclass, aminopenicillins, ampicillin and amoxicillin. Ampicillin available with the brand name Ampi and IUPAC name 2-amino-2-phenyl acetyl amino 3,3-dimethyl-7-oxo-4-thia, 1-aza-bicycloheptane 2-carboxylic acid. We have the structure of amoxicillin available with the brand name Amoxil and IUPAC name 6,2-amino-2,4-hydroxyphenyl acetyl amino 3,3-dimethyl-7-oxo-4-thia, 1-aza-bicycloheptane 2-carboxylic acid. Ampicillin and amoxicillin have similar spectrum of activity to penicillin gene but are more active against gram-negative cocci and enterobacteria. They are non-toxic and can be taken orally, but they are sensitive to beta-lactamase and are inactive against Pseudomonas aeruginosa. Some patients get diarrhea when they take penicillins. This is a result of poor absorption from the gut, with ampicillin being more poorly absorbed than amoxicillin. The alpha amino group plays an important role for broader activity. Amoxicillin is a para-hydroxy analog of ampicillin prepared by acylation of 6-amino-penicillinic acid with para-hydroxyphenylglycine. Amoxicillin is also called as beta-hydroxyampicillin, better absorbed through the gut wall due to presence of hydroxyl group. Same spectrum of activity as that of penicillin G, but more active against gram-negative bacteria, acid resistance hence given orally, non-toxic. Antibacterial spectrum is nearly identical with that of ampicillin, and like ampicillin, it is resistant to acid, sensitive to alkaline and beta-lactamase hydrolysis, and weakly protein bound. Amoxicillin is an antibiotic used to treat a number of bacterial infections. This includes middle ear infections, strep throat infections, pneumonia, skin infections, and urinary tract infections. Now moving on to carboxypenicillins, extended spectrum penicillins. We have examples here, the parent nucleus and substitution at R1 and R2. So we have carbenicillin, geopen, IUPAC name, substitution C6H5 and R at R1 and R2. Whereas carindacillin available with the brand name geocillin, R1, R2 substitution. Then ticarcillin, brand name is timantine and R1 and R2 substitutions. And carfacillin, that is available with the brand name uticillin, and both R1 and R2 are replaced by phenyl ring or substituted by phenyl ring. So IUPAC names are on the screen. Carbenicillin, as I told you, it is available with the brand name geopen. So it is a bactericidal antibiotic belonging to carboxypenicillin subgroup of penicillin. It is also more stable at lower pH than ampicillin. The antibiotic is highly soluble in water and is acid liable. It differs from ampicillin in having an ionizable carboxyl group rather than an amino group substituted on alpha carbon atom of the benzyl side chain. Carbenicillin was first penicillin to show activity against Pseudomonas aeruginosa. Compared with ampicillin, it is active against a wider range of gram-negative bacteria and was used against penicillin-resistant strains. The carboxylic group improves penetration of the molecule through the cell wall barriers of gram-negative bacilli compared with other penicillins, and it is used in high doses to cause bleeding. Carbenicillin at higher doses can cause bleeding. Use of carbenicillin can cause hypokalemia by promoting potassium loss at the distal convoluted tubule of the kidney. If we talk about ticarcillin, available with the brand name timantine, replacement with the group mentioned on the screen with R and R dash and IUPAC name mentioned on the screen. Ticarcillin is a alpha carboxy 3-thionenyl penicillin, an isoester of carbenicillin in which the phenyl group is replaced by thionenyl group. The semi-synthetic penicillin derivative like carbenicillin is unstable in acid and therefore must be administered parenterally. The advantages of ticarcillin is like it is slightly better pharmacokinetic properties including higher serum levels and a longer duration of action, and it is greater in vitro potency against several species of gram-negative bacilli and bacteroides fragilis. Moving on to uridocyclic urease penicillins, example piperacillin, mesalocillin, azolocillin, you can see on the screen. Basic nucleus and substitutions with the R group will get the different types of uridopenicillins. We have mesalocillin. So mesalocillin available with the brand name multocillin, and it is an acyl uridopenicillin with an antibacterial spectrum similar to that of carbenicillin, ticarcillin. We have piperacillin available with the brand name piperacil, and it is a broad spectrum beta-lactam antibiotic of uridopenicillin class. The chemical structure of piperacillin and other uridopenicillin incorporates a polar side chain that enables or enhances the penetration into gram-negative bacteria and reduces sensitivity to cleavage by gram-negative beta-lactamase enzymes. Piperacillin is most generally useful for extended spectrum acyl uridopenicillin. It is active more than mesalocillin against sensitive strains of gram-negative aerobic bacilli such as Serratia marcescens, Proteus, Enterobacter, Citrobacter species, and Pseudomonas aeruginosa. Piperacillin is destroyed rapidly by stomach acid, therefore it is active only by intramuscular or intravenous administrations. Now, talking about the structural activity relationship. So, position 1, you can see in screen when the sulfur atom of the thiazolidine ring is oxidized to a sulfone or sulfoxide, it improves the acid stability but decreases the activity of the agent. Position 2, no substitution allows at this position. Any change will lower the activity. The methyl groups are important here. The position 3, the carboxylic acid of the thiazolidine is required for activity. If it is changed to an alcohol or ester, activity is decreased. Position 4, the nitrogen atom here is very important, it is a must. Position 5, here no substitutions are allowed. Position 6, substitutions are allowed only on the side chain of the amide. An electron withdrawing group is added at this position, which will give the compound better acid stability because this substitution will make amide oxygen less nucleophilic. A bulky group added close to the ring will make the compound more resistant to beta-lactamases. Steric hindrance will protect or provide protection to the beta-lactam ring. Position 7, the carbonyl on the beta-lactam ring is must. And if you talk about the mechanism of action of penicillin, penicillin act by interfering with the development of cell wall, inhibit biosynthesis of dipeptidoglycan, acyclate, an inactive transpeptidase enzyme which cannot cross link linear peptidoglycan strands. Now moving on to the second class of beta-lactam antibiotic, that is cephalosporins. I would like to tell you about cephalosporins and few words that cephalosporins were discovered shortly after penicillin and entered into widespread product but not developed till 1960s. Cephalosporins are similar to penicillins but have a 6-member dihydrothiazine ring instead of 5-member thiazolidine ring. Okay, so together with penicillins and other drugs, it constitutes beta-lactam antibiotic. It possesses beta-lactam ring and act by same mechanism. Now, source of cephalosporin, it is obtained from Cephalosporium acremonium. It is the most important source of production of cephalosporin. It can be semi-synthetically prepared from 7-amino-cephalosporonic acid and 7-amino-3-deacetoxycephalosporonic acid. Structure and nomenclature. So, you can see on the screen, cephalosporins are a class of compounds which possess a bicyclic ring structure, and a ring A with a double bond unsaturation is termed as dihydrothiazine ring, while the adjoining ring, ring B, is termed as beta-lactam ring. So in penicillins you have seen the ring A was thiazolidine ring, and here it is dihydrothiazine ring. Thiazolidine ring in penicillin was 5-membered, and here dihydrothiazine ring is 6-membered, right? So both penicillins and cephalosporins shares the common beta-lactam ring. So it is same in both. Now numbering. Numbering of bicyclic ring starts with the nitrogen atom, and hence the sulfur atom in the dihydrothiazine ring is numbered as 5th. Chemically, cephalosporins are named as 5-thia-1-azabicyclo-oct-2-ene. Cepham is a name given to saturated bicyclic ring system. Cepham structure includes the carbonyl oxygen of the beta-lactam ring. You can see the Cepham nucleus. It is assigned to the bicyclic ring system which possesses a double bond at C3 position in dihydrothiazine ring. Now, classification of cephalosporins. Cephalosporins are classified into a series of generations. The first generation cephalosporins include ceftriaxone, cephalexin, cephaloglycine, cefazolin, cephaloridine. Now, one thing I would like to tell you that in penicillins we were adding selenes in the end, right? But in cephalosporins we will be adding cef as prefix. Their suffix was selene. Here the prefix is cef. So the first word or the first pronunciation or the first initiation will be cef here, and there it was selene. So we can remember the name, the second half of the name we can remember, and the first half will be same in pronunciation, and sometimes it is same as for all. The other drugs which includes in first generation cephalosporins are cephalothin, cefapyrin, cephadrine. The second generation cephalosporins are cefadroxil, cefprozil, loracarbev, cefamandole, cefonicid, ceforanide, cefmetazole, cefotictan, cefoxetine, cefuroxime. The third generation cephalosporins are cefdinir, cefixime, ceftibuten, cefoperazone, cefotaxime, ceftizidine, ceftriaxone, ceftriaxone. The fourth generation cephalosporins are cefepime, cefpyrone. Now the first generation cephalosporins. So we have this on the screen. You can see the different types of cephalosporins, their substitution on the basic nucleus, and the IUPAC names or different types of cephalosporins. So you can see on the screens. I can tell you the advantages of cephalosporins over penicillins. The cephalosporins have increased acid stability compared to penicillins. Most of the drugs here have better absorption than penicillins. They have broad antimicrobial spectrum. They have increased activity against resistant microorganisms. They have decreased allergenicity. they have increased tolerance than penicillins. So you can see on the screen the first generation cephalosporins, the different cephalosporins and their brand names and the substitution and IUPAC names. Cefadroxil, Duricef, available with the brand name and substitution and IUPAC names. Cephalexin, Keflex, substitutions and IUPAC names. Cephaloglycines, Kefglycine, substitutions and IUPAC name. Cefazolin, Ansef, substitution and IUPAC name. Cephaloridine, cephaloradine, and cephalothene, Cefalin, cefapyrin, cefadril, and cefadrine, Velocef. So different substitutions and IUPAC names. So it's easier to remember on a one slide. Now the second generation cephalosporins. So you can see the substitutions and different types of second generation cephalosporins. We have cefadroxil, Duricef, Ceprozil, Cefzil, substitutions and IUPAC name. Loracarbef, Lorabite, where CH3 instead of sulfur at C5 is replaced, and cefamandole, mandole, cefonicid, monocyt, ceforonide, Precef, cefmetazole, cefazone, cefotitan, cefotan, cefoxytin, that is mefoxin, cefuroxime, ceftin, and substitutions and IUPAC names. The third generation cephalosporins, again on the slide you can see the different types of third generation cephalosporins. Substitutions on R1 and R2 will give different types of third generation cephalosporins, their name and their brand names, and the IUPAC name is on the screen. Cefdinir, Omnicef, Cefixime, Suprax, Ceftibutin, Sidax, Cefoperazone, Puricef, Cefotaxime, Claroforan, Ceftazidime, Fortas, Ceftizoxime, Cefizox, Ceftriaxone, Rocephin. So substitutions and IUPAC names. We have fourth generation cephalosporins. Examples, two cephalosporins which comes under fourth generation cephalosporins. That is Cefepime, IUPAC name is mentioned on the screen, and substitutions, brand name Maxipime, Cefpyrone, brand name Cephir, and substitutions and IUPAC name. Now structural activity relationships of cephalosporin. So you can see there is 7 acyl amino modification. So here the amino group acylation increases gram positive activity and decreases gram negative activity. Attachment or replacement of any other heterocyclic in side chain increases gram positive activity. Catechol group in side chain increases the activity, and lipophilicity of the side chain increases gram negative activity. Aminothiazole group increases gram negative activity and resists beta-lactamase in combination with syn-oxyamino grouping. Third carbon substituent modification. So here modification at third carbon position reduces degradation. Ester amide nitrogen nucleophile, sulfur nucleophile at third carbon increases pharmacokinetic properties. Replacement of third acetoxy group by aromatic thiols results in increase in activity against gram-negative bacteria. Replacement of 3-acetoxy group by chloro group gives good oral absorption. Seventh carbon substituent modification, where 7th carbon alpha pharma amidino derivatives are good for high beta-lactamase stability. Ring modification, oxidation of sulfur to sulfoxide or sulfone increases the activity against beta-lactamase enzyme. Replacement of sulfur by oxygen increases activity. And the fourth carboxylic acid group modification, the fourth carboxylic acid group to ester increases the bioavailability. C3-C4 linkage is important for the activity. Isomerization of second and third position leads to loss of activity. Mechanism of action will be same as that of penicillins. Now let us talk about the comparison of 6-amino penicillinic acid and 7-amino cephalosporonic acid. I hope you remember this both, because these are the basic nucleases for penicillins and cephalosporins. So you can see on the screen, we have categorized them, we are differentiating them, we are comparing them. This is most important, as from examination point of view, you may definitely get question from this. Synonym 6-amino penicillinic acid, 7-amino cephalosporinic acid. Structures are mentioned, where ring A is thiazolidine ring, 5-membered ring in penicillins, whereas in cephalosporinic acid or cephalosporins, ring A is dihydrothiazine ring. It is a 5-membered ring in penicillin, whereas it is a 6-membered ring in cephalosporins. Both ring B will be beta-lactam in both, and ring A is devoid of double bond in penicillins, whereas a ring A contains double bond in between second and third carbon in cephalosporins. Nomenclature, IUPAC name, you can see molecular formula is on the screen. Isolation, how it is obtained. The 6-aminopenicillinic acid is obtained isolated from cultures by addition of acylases, whereas 7-aminocephalosporinic acids are obtained from hydrolysis of cephalosporin C. Hydrolysis, preparation you can see, and then you can see the stability. Penicillins will have less stability towards acids, whereas 7-aminocephalosporinic acid will have more acid stability. We also compare penam and cefam. Penams in penicillins are primary skeleton structure that defines the penicillin subclass of broader beta-lactam family of antibiotics and related compounds, whereas in cefam, cephalosporins, cefam is an organic heterobicyclic compound that consists of 5-thia-1-aza bicyclooctane bearing an 8-keto substituent. The parent of the class of cefam, natural fundamental product parent, a member of cefam and is saturated organic heterobicyclic parent. So you can see again ring A and ring B. Ring A is again 5-membered thiazolidine ring in penam, whereas in cefam it is 6-membered dihydrothiazine ring. Both has ring B as beta-lactam ring, and molecular formula, nomenclature, molecular weight. Basic skeleton for penicillins, it is a basic skeleton for cephalosporin, cefam. It is termed as penams when double bond is present in ring A between second and third carbon. It is termed as penam, whereas it is termed as cefam when double bond is present in ring A between second and third carbon. It is very important to note here commercially available cephalosporins including cefamycins will have cefam nucleus. Now we have third beta-lactam antibiotic, that is carbapenem. These are the class of beta-lactam antibiotic that are active against many aerobic and anaerobic gram-positive and gram-negative organisms. So you can see the IUPAC name and structure on the screen. Example for carbapenem is thienamycin, which was first discovered in 1976. Then we have monobactam, and these are the subgroup of beta-lactam antibiotic wherein the beta-lactam ring is alone and not fused to another ring, in contrast to the other beta-lactam antibiotics which have at least 2 rings. Monobactam work only against aerobic gram-negative bacteria like Neisseria, Pseudomonas, etc. Now moving on to the second class of antibiotic, that is aminoglycosides, and aminoglycosides consist of amino sugar linked to a central highly substituted aminocyclitol ring by means of glycosidic linkages. So they are called as aminocyclitol antibiotics. So the aminocyclitol ring is replaced by a streptidine ring in streptomycin and dihydrostreptomycin. Source is like it is obtained from actinomycetes bacteria belonging to the genus Streptomyces. Some of these antibiotics can be semi-synthetically derived from naturally occurring aminoglycosides. They consist of 2 or more amino sugars joined in glycoside linkage to a highly substituted 1,3-diaminocyclohexane centrally placed ring. You can see on the structure on the screen. The ring is 2-deoxystreptamine in all aminoglycosides except streptomycin, where it is streptidine. Now examples here are kanamycin, 2 amino sugars are attached to 2-deoxystreptamine. Streptomycin, in streptomycin 2 amino sugars are attached to streptidine, and in neomycin 3 amino sugars are attached to 2-deoxystreptamine. Now in short, aminoglycosides contain 2 important features. One is amino sugar portion and second is central hexose ring. That is either 2-deoxystreptamine or streptidine. These series include streptomycin, neomycin, kanamycin and gentamicin, and the structures are on the screen. The first one, you can see it's streptomycin available with the brand name Ambistrine. Streptomycin produced by a culture called Streptomyces graecosus, and on acid hydrolysis, streptomycin yields streptidine and streptobiosamine. That is a combination of L-streptose and N-methyl L-glucosamine. IUPAC name is mentioned on the screen, so you can see. Next compound is gentamicin, that is garamycin. So gentamicin is systematically administered aminoglycoside antibiotic to be introduced for clinical use and was obtained from Micromonospora purpurea. So you can see there are different types of gentamicin, C1, C2, C1a and C2a. You can see the replacement of R in the group or in the structure. So you will get the different types of garamycins. IUPAC name is again on the screen. SAR of aminoglycosides. So first amino sugar portion, that is C6 and C2, serves as a major target for bacterial inactivation. Methylation at 6th position does not affect activity but increases enzyme resistance. Methylation at 6th position does not affect activity but increases enzyme resistance. Cleavage of 3-hydroxyl or 4-hydroxyl or both does not affect the activity. The ring is important for broad spectrum activity and responsible for inactivation of bacterial enzyme. So we have next product, that is neomycin, available with the brand name Neofradine. Neomycin B and C is also available, and you can see the substitution at R1 and R2, and whereas ring 1, 2, 3, 4, we have different names for the ring. Ring 1 is neosamine C, 2 is 2-deoxystreptamine, 3 is D-ribose, and 4th is neomycin B and C, where it is based on R1 and R2. So neomycin is isolated from Streptomyces fradii. Neomycin is active against gram-negative bacilli and some gram-positive cocci. Pseudomonas and Streptococcus pyogenes are not sensitive here with neomycin. Neomycin is highly toxic to internal ear, that is mainly auditory, and to kidney. So it is therefore not used systematically. Absorption from the GIT is minimal, so oral and topical administration does not ordinarily cause systematic toxicity. Now UPAC name is on the screen: 5-amino-2-amino-methyl, 6,4,6-diamino, 2,4,3-amino-6-amino-methyl, 4,5-dihydroxy, oxan-2-yl, oxy, 3-hydroxy, 5-hydroxy-methyl, oxalan-2-yl, oxy, 3-hydroxy-cyclohexyl, oxyoxane, 3,4-dioyl sulfuric acid. We have kanamycin. Kanamycin is available with the brand name Cantrax, and you can see the substitution R1 and R2 in the structure. The ring 1 is 6-amino 6-deoxy D-glucose, ring 2 is 2,6-diamino 2,6-deoxy D-glucose, and ring 3 is 2-amino 2-deoxy D-glucose, where we can substitute R1 and R2 to get different types of kanamycin A, B, C, IUPAC name on the screen. So kanamycin is isolated from Streptomyces kanamyceticus, and it is active against Mycobacterium and mainly intestinal bacteria. Kanamycin A, B, C are commercially available, and most pure form is kanamycin A and least toxic of all three forms. The kanamycin differs only in the sugar moiety attached to glycosidic oxygen on the 4th position of central deoxystreptamine. Kanamycin A contains 6-amino 6-deoxy D-glucose. As I told you, kanamycin B contains 2,6-diamino 2,6-deoxy D-glucose, and kanamycin C contains 2-amino 2-deoxy D-glucose. The mechanism of action of aminoglycosides: they bind to 30S ribosomal subunits and interfere with initiation of protein synthesis by fixing 30S and 50S ribosomal complex at the starting codon AUG of mRNA, where further protein synthesis will not occur. They also cause misreading of mRNA, leading to premature termination of translation with detachment of ribosomal complex and incomplete synthesized protein. Introduces abnormal codon on mRNA that leads to abnormal attachment of amino acid and abnormal protein synthesis occurs. Now, if you talk about the structural activity relationship of aminoglycosides, so central hexose ring, first carbon amino group modification like acylation in ethylation helps in retaining the potency, whereas second hydroxylation and fifth deoxygenation increases inhibition of bacterial inactivating enzyme system. Second amino sugar position, we have replacement of 2-hydroxyl group by amino group, which may increase the activity, and replacement of third carbon by secondary and tertiary amine for primary amine increases the activity. Now, the third class of antibiotics, that is tetracycline, the name itself says tetra 4-cycle cyclines. These class of potent broad spectrum antibiotics derive from the culture of Streptomyces bacteria. Source is like naturally they are obtained from various species of Streptomyces bacteria by fermentation technology. These are also obtained semi-synthetically from naturally obtained tetracyclines. Tetracyclines are considered as broad spectrum antibiotics. They are the derivatives of octohydronaphthacine. Basic nucleus you can see on the screen. It has 4 rings, 1, 2, 3, 4, and it is called as naphthacene carboxamide, and it possesses 4, 4A, 5A, 6 and 12A potentially asymmetric carbon atoms. Oxytetracycline and doxycycline have 6 asymmetric centers, whereas the other contains only 5 asymmetric centers. Structures you can see on the screen. There are nearly 5 types of tetracycline. Basic nucleus is on the screen, and substitution R1, R2, R3, R4 will give different types of tetracyclines. IUPAC names are mentioned. Tetracycline available with the brand name Somycin. Chlortetracycline available with the brand name Aureomycin, oxytetracycline, terramycin, doxycycline, adoxa, and minocycline, dinosine. So tetracycline available with the brand name Somycin, and it is stable in acid solution with a pH about 2. It is used to treat a number of infections including acne, cholera, brucellosis, plague, malaria, and syphilis. Chlortetracycline available with the brand name Aureomycin and substitution R1, R2, R3, R4, and IUPAC name is mentioned on the screen. Oxytetracycline available with the brand name terramycin, and it is a pale yellow beta crystalline compound. The amphoteric base is only slightly soluble in water and slightly soluble in alcohol. Doxycycline available with the brand name Odoxa. It is 6-deoxy-5-oxytetracycline. Absence of 6-hydroxyl group produces a compound that is very stable in acids and bases and that has a long biological half-life. Minocycline available with the brand name Dinosine. So it is the most potent tetracycline currently used in therapy, and it is obtained by a reductive methylation of 7-nitro-6-demethyl-6-deoxytetracycline. Because minocycline, like doxycycline, lacks the 6-hydroxyl group, it is stable in acids and does not dehydrate or rearrange to anhydro or lactone forms. Now, structural activity relationship of tetracyclines: the carbon system present at first and to third must be firm for good activity. The amide group at second position is important for the activity, and substitution at sixth position will decrease the chemical stability. Sixth carbon methylated analogues will have high blood levels, and seventh carbon substitution leads to increased potency. 6-hydroxyl group and 5-alpha hydrogen leads to dehydration by strong acids, which leads to development of 6-deoxytetracyclines. At 12th alpha position, cis-type fusion between A, B with hydroxyl group is important for withholding of activity. At 7th position, electron donating and withdrawing groups are recommended, and position 5th, 6th, 7th and 9th can be modified with various substituents for improvement in the activity. Sulfur atom at 6th position leads to thiotetracycline, shows excellent pattern of activity. Amino alkylation at carboxamido group will increase water solubility, and semi-synthetic analogues for treatment of neoplasm are synthesized. Example: methicycline, doxycycline, minocycline. Mechanism of action: tetracycline acts by binding to 30th subunit of ribosome at the A site. During protein biosynthesis, the new tRNA with the amino acid attempts to bind the A site of the ribosome. However, since A site is blocked by tetracycline, the amino acyl tRNA cannot bind to it. Thus, without sequential attachment of tRNA at the A site, protein biosynthesis cannot occur. By inhibiting protein biosynthesis, tetracyclines cause cell death of the bacterial cell. Now, the fourth class of antibiotic is macrolide antibiotics, and macrolides have three general chemical characteristics. One is lactone ring, second is many ketonic and hydroxyl functions, third is glycosidically linked 6-deoxy sugar, and the lactone ring has 12 or 14 or 16 atoms in it, and it is often unsaturated with an olefinic compound or group conjugated with the ketone functions. They have many neutral sugars along with the amino sugar that is linked glycosidically to the lactone ring. If we talk about the classification of macrolide antibiotic, they are classified according to the number of lactone ring, like 12-membered ring molecule, 13-membered tulathromycin, 14-membered. Most drugs are from this category. Erythromycin, 15-membered, 16-membered. Again, the most drugs are from this category. Spiramycin, tylosin, tilmicocin. Along with the above, some special groups are also available, like azelides, 15-membered erythromycin, triamylides, example combination of 13 and 15-membered tulathromycin, and third is ketolides, 14-membered with ketonic group, telithromycin. Mechanism of action of macrolides revolve around their ability to bind the bacterial 50S ribosomal subunit, causing the cessation of bacterial protein synthesis. Structure: we have erythromycin available with the brand name bristamycin, and structure is on the screen and IUPAC is mentioned below. Erythromycin, a 14-membered, is a naturally occurring macrolide derived from Streptomyces erythraeus. There is a problem with acid liability, narrow spectrum, poor gastrointestinal tract absorbance or intolerance, short elimination or half-life. Structural derivatives obtained from this erythromycin: we have newer macrolides, which includes clarithromycin, oxythromycin, azithromycin. Neomacrolides, we have broader spectrum of activity, improved pharmacokinetic properties, better bioavailability, better tissue penetration, prolonged half-lives, that is improved tolerability. The amino sugar attached through a glycosidic linkage to fifth carbon is desosamine, a structure found in several other macrolide antibiotics. Erythromycin can be used as a free base in oral dosage form and for topical administration to overcome its bitterness and irregular oral absorption resulting from acid destruction and adsorption onto food. Several enteric-coated and delayed release dose forms of erythromycin base have been developed. These forms are fully successful in overcoming the bitterness but have solved only marginal problems of oral absorption. Erythromycin is chemically modified with primary two different aims to increase either its water or its lipid solubility for parenteral dosage form or to increase acid stability and for improved oral absorption. We have another drug that is clarithromycin available with the brand name Biclar and APAC name and structure is on the screen. Clarithromycin is a 6-methyl ether of erythromycin. The simple methylation of 6-hydroxyl group of erythromycin creates a semi-synthetic derivative that fully retains antibacterial property for parent antibiotic. Clarithromycin is a 6-methyl ether of erythromycin. The simple methylation of 6-hydroxyl group of erythromycin creates a semi-synthetic derivative called clarithromycin that fully retains the antibacterial properties of the parent antibiotic with marked increased acid stability and oral bioavailability and gastrointestinal side effects. We have next drug that is roxithromycin available with the brand name Roxibid and impact name and structure is on the screen. Roxithromycin is semi-synthetic macrolide antibiotic. It is used to treat respiratory tract, urinary and some tissue infections. Roxithromycin is derived from erythromycin containing the same 14-membered lactone ring. However, an N-oxime side chain is attached to the lactone ring. It is semi-synthetically longer acting, acid stable and good enteral absorption. We have azithromycin, azithral, IUPAC name and structure is on the screen. Azithromycin is a semi-synthetic derivative of erythromycin prepared by Beckmann rearrangement of the corresponding 6-oxime followed by N-methylation and reduction of the resulting 6-ring expanded lactam. It is a prototype of a series of nitrogen-containing 15-membered ring macrolide known as azalide. Removal of the 9-keto group coupled with incorporation of the weakly basic tertiary amine nitrogen functions into macrolide ring, increasing the stability of azithromycin to acid catalyzed degradation. This changes also increases the lipid solubility of the molecule, thereby conferring unique pharmacokinetic and microbial properties. Structural activity relationship of macrolide antibiotics, variation of substituents on pyrrolidine portion and fifth carbon side chain affects the activity. And demethylation imparts activity against gram-negative bacteria, increasing chain length of the propyl substituent at 4th position in pyrrolidine moiety up to N-hexyl increases in vivo activity about 1.5 times than parent compound. The thiomethyl ether of alpha-thio lincosamide moiety is essential for the activity. Structural modification at C7 like introduction of 7-chloro or 7-R methoxy changes the physicochemical parameters of the drug, that is partition coefficient, and thus alters activity spectrum and pharmacokinetic properties. Mechanism of action, macrolide antibiotic, as I told you before, they are the bacteriostatic agents that inhibits protein synthesis by binding irreversibly to a site on the 50th subunit of the bacterial ribosome, thus inhibits the translocation step of protein synthesis. Now moving on to the fifth class of antibiotics, that is lincosamide antibiotics. These are the antibiotics obtained isolated from actinomycetes, streptomyces, lincolnensis. They bind to 50th ribosomal subunit to inhibit protein synthesis, and action may be bactericidal or bacteriostatic depending on many factors like concentrations of antibiotic. Structure, lincomycin available with the brand name Lincocin. You can see the UPAC name on the screen, and lincomycin structure contains a basic function, the pyrrolidine nitrogen, by which water soluble salts with an apparent pK of 7.5 may be formed. It has an antibacterial spectrum and mechanism of action similar to macrolides. Lincomycin is also effective against organisms including actinomycetes and some species of Mycoplasma and Plasmodium. We have another compound, clindamycin, available with the brand name Cleocin, and structure and UPAC name is on the screen. Clindamycin sulfam palmitate hydrochloride formulation is the hydrochloride salt of the palmitic acid ester of cliomycin. The ester bond is to the 2-hydroxyl group of the lincosamine sugar. The ester serves as a tasteless product form of the antibiotic which hydrolyzes to clindamycin in the plasma. Clindamycin is used in the treatment of number of bacterial infections including bone or joint infection, pelvic inflammatory disease, throat infection, pneumonia, middle ear infection and endocarditis. It can also be used to treat acne and some cases of methicillin resistant staphylococcus aureus. Side effects may include rashes, urticaria and abdominal pain. Next class is polyene antibiotic, which are also called as polyene antimycotics, polyene compounds that target fungi. These polyene antimycotics are typically obtained from species of Streptomyces bacteria. The polyene binds to ergosterol in the fungal cell membrane and thus weakens it. It causes leakage of potassium and sodium ion which may contribute to fungal cell death. Amphotericin B, nystatin and natamycins are the examples of polyene antimycotics. Structures, we have amphotericin B which is available with the brand name Fungizone and UPAC name is on the screen. So we have the second drug nystatin, that is Nilstat. Structure and UPAC name is on the screen. So we have another drug that is called as natamycin available with the brand name Natacin. Structure and UPAC name is on the screen. We have another class, that is second last subclass, that is polypeptide antibiotics. So these are the antibiotics, chemically diverse class of anti-infective and anti-tumor antibiotics containing non-protein polypeptide chains. Examples here include bacitracin, polymyxin B, colistin, vancomycin and rifamycin. Source for this member is the class of drugs obtained from Bacillus species such as Bacillus subtilis, that is bacitracin, Bacillus polymyxa, that is polymyxins, and Bacillus brevis, that is gramicidins. Bacitracin, the first compound, the structure is on the screen. Then comes polymyxin, structure is on the screen and UPAC name is on the screen, obtained from Bacillus polymyxa. At least 5 polymyxins are available, that is A, B, C, D and E. Only polymyxin B and E are of clinical importance. Polymyxins are biosynthesized by bacteria belonging to Gram Bacillus polymyxa. As I told you, more specifically, the subgenus Penibacillus. Polymyxins consist of 10 amino acid residues, 6 of which are L-alpha gamma diaminobutyric acid. The diaminobutyric acid residues cause polymyxins to have multiple positively charged groups at physiological pH. 7 amino acid residue forms the main cyclic components, while the other 3 extend from one of the cyclic residue as a linear chain terminating in either 6-methyl octanoic acid or 6-methyl heptanoic acid at the end terminus. Polymyxin A, C and D are too toxic for human use. So polymyxins act on bacteria via disrupting the transport mechanism of the cell membrane. The application of polymyxin to treat serious cases of infection caused by Pseudomonas aeruginosa strain is rare. Next is colistin. So colistin available with the UPAC name as on screen and structure is on the screen. It is isolated as an antibiotic from Aerobacillus clostinus that was given the name colistin. It is cationically cyclic decapeptide linked to a fatty acid chain through an alpha amide linkage amino acid, that is D-leucine, L-threonine, L-alpha gamma diaminobutyric fatty acid. Could be 6-methyl octanoic acid, 6-methyl heptanoic acid. 6-methyl octanoic acid as colistin A and 6-methyl heptanoic acid as colistin B. Colistin A, which differs from polymyxin B only by substitution of D-leucine for diuphenyl alanine as one of the amino acid fragments in the cyclic portion of the structure. 2 forms of colistin have been prepared, the sulfate and methane sulfonates. So moving on to vancomycin. Vancomycin structure is on the screen, is isolated from Streptomyces orientalis. It is discovered in 1956 as penicillin substitute, which assumes specific significance due to efficacy against methicillin resistant Staphylococcus aureus, viridans and Enterococcus. Bactericidal action is exerted on gram-positive cocci, Neisseria, Clostridia and diphtheroids. Vancomycin acts by inhibiting bacterial cell wall synthesis. It binds to the terminal dipeptide D-ala sequence of peptidoglycan units, prevents its release from bactoprenol lipid carriers so that assembly of the units at the cell membrane and the cross-linking to form the cell wall cannot take place. Common side effects include pain in the area of injection and allergic reactions. Occasionally, hearing loss, low blood pressure, or bone marrow suppression occurs. Next is rifamycin. So rifamycin B, rifamycin SV is on the screen. So these are useful in treating tuberculosis, leprosy, and mycobacterium, avium complex infections. Rifamycin A, B, C, D, E, S, and SV is available. Structural activity relationships. So rifampicin is formed by substitution of 4-methyl 1-piperazine aminomethyl at third position of rifamycin B. It is a bactericidal antibiotic of rifamycin group and it is orally active. Rifapentine compound is formed by substitution of 4-methyl 1-piperazine aminocyclopentane at third position of rifamycin B and it is more potent than rifampicin. Now hydroxyl group at first carbon, 8th carbon, 21 carbon and 23rd carbon all lie in a plane and play a crucial role in acting as binding groups for attachment to DNA dependent RNA polymerase. Activity of compounds get diminished when the hydroxyl group at 2nd carbon, 23rd carbon are acetylated. Both reductions of double bond in macrocyclic as well as its ring opening results in compounds with decreased activity. These modification greatly affects rifamycin's conformational structure which decreases its ability to bind to DNA dependent RNA polymerase. Modification at 3rd and 4th carbon atom results in compounds having antibacterial property of different capacities, also affects their transportation across the bacterial cell membrane. Mechanism of action, they damage the integrity of cytoplasmic membrane by disorienting the lipophilic group present in the membrane, which leads to leakage of intracellular components. They combine with the membrane sterols and thus creates pores or channels in the fungal membrane. Now moving on to the last class, that is miscellaneous antibiotics, we have example chloramphenicol. It is isolated from Streptomyces venezuelae and the structure is on the screen. It is less active against gram-positive bacteria when compared to tetracyclines. Chloramphenicol possesses 2 chiral carbon atoms in acyl amido propene diol chain. Biological activity resides almost exclusively in the D-thrio isomer L-thrio and D and L erythroisomers are virtually inactive. Strains of certain bacterial species are resistant to chloramphenicol by virtue of the ability to produce chloramphenicol acetyl transferase, an enzyme that acetylates the hydroxy group of the position 1 and 3. Both the 3-acetoxy and 1,3-diacetoxy metabolites lack antibacterial activity. The mechanism of action here is chloramphenicol inhibit protein synthesis in bacteria and to a lower extent in eukaryotic cells. The drug readily penetrates bacterial cells, probably by facilitated diffusion. It acts primarily by binding reversibly to 50S ribosomal subunit, although binding to tRNA at a codon recognition site on 30S ribosomal subunit is undisturbed. The drug appears to prevent the binding of amino acid containing end of the amino acyl tRNA to acceptor site on the 50S ribosomal subunit. The interaction between peptidyl transferase and its amino acid substrate cannot occur and peptide bond formation is inhibited. If we talk about the structural activity relationship of chloramphenicol, paranitrophenol group is present there, you can see on the screen. Replacement of this para-nitro group by other substituent will lead to reduction in the activity. Shifting of the nitro group to para position also reduces the antibacterial activity, whereas the replacement of phenyl group by alicyclic molecule results in less potent compounds. The para-nitrophenol group may be replaced by other aryl structures without appreciable loss of activity. The dichloroacetamide side chain. Other dihalo derivatives of side chain are less potent, through the major activities are retained, while in case of trihalo derivatives, the 2-NHCOCF3 derivatives would be about 1.7 times as active as chloramphenicol. 1,3-propanediol, so the primary alcoholic group on first carbon, if modified, results in decrease in activity, hence the alcoholic group is to be considered as very important for the activity. Of the 4 stereoisomers of chloramphenicol, the antibacterial activity resides in only D-thrio compound. Other isomers are inactive compounds. Now we have other antibiotics which comes under the classification of miscellaneous antibiotic. Example is novobiocin, available with the brand name albamycin. Structure and impact name is on the screen. Novobiocin is available as novobiocin sodium and it is anti-coumarin antibiotic produced by Actinomycetes streptomyces nivens with antibacterial property. No biosine as well as other aminocoumarin antibiotics inhibit bacterial DNA synthesis by targeting at the bacteria DNA gyrase and related enzyme DNA topoisomerase 4. We have another compound, mupirocin. Bactroban is a brand name. So mupirocin structure and impact name you can see on the screen, and it is used as ointment to treat impetigo and secondary skin infection caused by Staphylococcus aureus and Staphylococcus pyogenes. It is termed pseudomonic acid A, and it is a novel antibiotic agent with a unique chemical structure and mode of action apart from other antibiotic agents. Now let us move on to the synthesis of ampicillin. So ampicillin is synthesized by taking phenylglycine reacted with ethyl chloroformate in presence of triethylamine to give an intermediate called as phenylglycine anhydride with monoethyl carbonate, which again reacted with 6-aminopenicillinic acid in presence of sodium bicarbonate, giving an sodium salt of ampicillin. Now if you talk about the synthesis of amoxicillin, then amoxicillin is synthesized by taking Dane salt, which is sodium alpha 4 hydroxyphenyl alpha 2 methoxycarbonyl 1 methyl ethinyl amino acetate, reacting with ethyl chloroformate in presence of N-methyl morpholine to give an intermediate which in reaction with 6-aminopenicillinic acid in presence of trimethyl chlorosilane at pH of 1.1 to 1.2 will give amoxicillin. Now moving on to the synthesis of cephalexin. So it is synthesized or obtained by taking phenylglycine and reacted with acetyl anhydride in presence of pyridine and removal of hydrochloride molecule. It give an intermediate which again in presence of CHCOCl, CHT twice will give intermediate which again reacted with 6-aminopenicillinic acid and in presence of trifluoroacetic acid will give cephaloglycine, which on dehydrogenation will give cephalexin. Now moving on to the synthesis of cephadroxil. Cephadroxil can be prepared in 3 steps. In step 1, 7-amino desacetoxycephalosporinic acid is taken and this is reacted and undergoes sialylation in presence of hexamethyl disilazane trimethyl chlorosilane in dichloromethane to give product A. In step 2, Dane salt is taken and this is reacted with ethyl chloroformate and N-methyl morpholine to give product B. In step T, this A and product B both are taken and that are subjected to N-acylation and hydrolysis to give cephadroxyl carbonate. If we talk about the synthesis of oxytetracycline, the biosynthesis of oxytetracycline begins with the utilization of pKase enzyme ketosynthase, the chain length factor, the acyl carrier protein and an acyl transferase to catalyze the extension of malonamyl CoA starting unit with 8 malonyl CoA extender units. The process of elongating the polypeptide skeleton occurs through a series of Claisen light decarboxylation reactions until the linear tetracycline skeleton is formed. Thus, minimal pKa's form a completed amidated polyketide backbone without any additional post-synthase tailoring enzymes. Following the formation of linear tetracycline skeleton, 4 successive cyclization reactions must occur in a regioselective manner to produce the aromatic natural product known as pre-tetramide, a common precursor to both oxytetracycline and other tetracycline antibiotics. In the oxytetracycline gene cluster, these enzymes are encoded as oxy-K aromatase, oxy-N cyclase and oxyl, that is cyclase. Formation of pre-tetramide allows for one of the most important intermediates en route to the biosynthesis of tetracyclines. If we talk about the synthesis of doxycycline, it is synthesized from oxytetracycline by dehydrating oxytetracycline at 6th carbon by reducing the tertiary hydroxyl group with hydrogen using a rhodium on carbon catalyst. So this completes antibiotics.